Today we will discuss structure and reproduction of viroids. In 1971, Theodore O. Diner, a pathologist working at the Agriculture Research Service, discovered an acellular particle that he named a viroid, meaning virus-like. The first viroid discovered was found to cause potato tuber spindle disease, which causes slower sprouting and various deformities in in potato plants. Viroids are an independent class of plant pathogens which are distinguished from viruses by number one, the absence of a protein coat, and second, by their unusually small size. A viroid is 80 times smaller than a virus. Viroids consist of single-stranded covalently closed circular RNA in both rod shape and branch structures. They are only 250 to 400 bases long. A simple definition of viroid is viroids are small, circular, single-stranded, non-protein coding RNAs that replicate autonomously when inoculated into higher plants. Some are pathogenic, but others replicate without eliciting symptoms in susceptible plant species. Unlike the genomes of conventional viruses, viroids are not protected by a protein capsid. Next is structure of viroids. Viroids consist only of naked RNA without any protective protein coat. Their genomes are small, single-stranded circles of RNA that are only 250 to 400 bases long. They have no protective coat, as I already told you, just single-stranded RNA. You can see here the structure of coconut kedeng viroids showing complete sequence, means 246 bases of coconut kedeng viroid. Although viroid RNA is single-stranded, but base pairing occurs between the bases on the opposite halves of the circle to produce a rod-like structure. I can see here in this diagram, single-stranded circle, but base pairing occurs between the halves on the on the opposite halves of the circle to produce a rod-like structure. This is rod-like structure, and this is branched viroid structure showing branches. So because viroids have no protein coat, they lack attachment proteins and cannot recognize and penetrate healthy cells as can a virus, true virus. So viroids can infiltrate a plant cell only when its surrounding membrane is already damaged. So they often take advantage of a damage done to plant tissues by insects. Once inside, viroids may be means passed from one plant cell to another via cellular junctions. Next is classification of viroids. Currently, 33 different viroid species and several sequence variants have been biologically and molecularly characterized. Based on structure characteristic and their impact on biological properties, viroids have been classified by International Committee on Virus Taxonomy, that is ICTV, into two families. One is Pospiviridae and the second is Epson viroidae family. You can see here family Epson viroidae having 3 genera and 4 species. Genus Epson viroid, then Palmoviroid and Elaviroid. First genus having 1 species, that is avocado sunblotch viroid. Genus Palmoviroid having 2 species, Chrysanthemum chlorotic mottled viroid and peach latent mosaic viroid. Similarly, third genus, that is Elaviroid, having 1 species, eggplant latent viroid. Second family, Pospiviridae, having 5 genera and 28 different species. Genus Pospiviroid having 10 species, genus Hostiviroid having 1 species, and genus Viroid having 4 species. Genus Epsca viroid 10 species, and genus Viroid having 3 species. Next is reproduction or multiplication of viroids. Viroids replicate by rolling circle mechanism. Viroid RNA does not contain any gene that encode proteins. It merely carries signal for its own replication by the host machinery. Although viroid encodes no protein enzyme, the viroid RNA itself acts as a ribozyme. That is, the RNA catalyzes an enzymatic reactions. Most viroids replicate in the plant cell nucleus and rely on the RNA polymerase 2 for RNA synthesis. A smaller group of viroids, for example, chrysanthemum chlorotic mottle viroid, have a highly branched structure rather than a rod with bulges and replicate in the chloroplast. The viroid own ribozyme activities are used for self-cleavage of the multimeric RNA generated during replication. So host enzymes provide all other functions. So different steps in the multiplication of viroid. First of all, you can see here infection viroid RNA. First, host RNA polymerase copies the circular plus strand to form multimeric minus strand. So this is plus strand, synthesis of negative strand, right? So second next step is site-specific cleavage by viroid ribozyme. So site-specific cleavage of this strand by viroid ribozyme gives monomeric that are circularized by the host RNA ligase, right? Circularization of this by host RNA ligase. Next, this minus stranded circles are then the templates for the second round of rolling circle replication by RNA polymerase, means synthesis of positive strand again, and the resulting multimeric plus strand undergoes ribosome cleavage. This undergoes cleavage to create monomers. So these are again circularized to produce the progeny viroid, means circular positive single stranded RNA. Thanks for watching.